Hello students, this is Vedant Mirza. We are discussing transport in plants. When we're discussing transport in plants, there are some basic facts that we have to keep in mind. Like why there is a need for transport system in plants. There is no circulatory system like the cardiovascular system that we have in our bodies. But plants do have a transport system. In fact, two major transport systems, the xylems and the phloems. Why do they need transport systems? Plants need transport system to move the substances from where they are produced to where they are needed. And these are needed for metabolic reactions. Metabolic reactions include all the chemical reactions, the sum total of chemical reactions occurring in the cells. Water, iron, glucose are required for that. For growth, these substances are required. So the glucose can be delivered to all the growing parts and mitosis can proceed, and if required, meiosis as well. For storage as well, so glucose can be stored as starch. These are the main points that one should keep in mind why transport system is actually required in plants. Gases also need to be transported, oxygen, carbon dioxide. These are not transported through the transport system though, unlike our bodies, unlike the mammalian transport system. The air spaces in leaf, stems and roots, there are plenty of them, and they allow diffusion of these gases into the tissues and into each of the cells. Leaf, stem, roots, they are all organs and they are made up of various tissues. And all these tissues, they gain these gases by diffusion alone. Stomata is the main part of the leaf through which gaseous exchange occur, where the air inside the leaf is in continuity with the outer environment. Two major transport systems which are there in plants, they work together. They work to ensure that plant is delivered with water, salts as well as the sucrose and amino acids. Xylem, it transports water and mineral ions. Phloem, on the other hand, it transports sucrose and amino acids. In xylem, the movement is from roots to leaf, which is always unidirectional. In phloem, it's always either from leaf to other parts of the plants, where it could either be used or stored, which is again unidirectional. But the unidirectional flow in phloem is in one direction in one phloem, but it can be in the other direction in the other phloem, which means that, for example, if a phloem is bringing the stored starch in the form of sucrose back to the aerial parts of the plant, for example, growing tips, then in that phloem, the flow will be against the direction in which it actually went from the leaf to the root. So in phloem, the direction can be bidirection, but it will always be unidirection for one phloem at one time. To understand how the plants, especially the flowering plants which we are to discuss, they are working, we have to follow the structure of stem, the structure of roots and the leaves. And to understand these, the tissues which are present in these structures, these tissues need to be understood as well. The placement of these cells, where they are present, what do they do, where you can label them. Here I am listing down the major tissues which are we to discuss in this chapter. For these tissues, you must remember the labels on various diagrams which will be given to you. Epidermis, parenchyma, collenchyma, endodermis, mesophyll, pericycle. And then there's vascular tissues, xylem and phloem. Xylem includes the xylem vessel elements and sclerenchyma. And phloem, it includes the sieve elements and the companion cells. So these are the major parts of the you can say the tissues that you have to keep in mind. Epidermis, let's start these one by one. So we are discussing epidermis here. Epidermis is a continuous layer of one cell thick lining on the outer side of the plant. It's always one cell thick. It's transparent in most cases. It's having a waxy cuticle that covers this layer in leaf and stem. It provides waterproofing. It prevents water loss and drying out as well as infection. It doesn't allow the pathogen to penetrate. So if this is the epidermis, one cell thick, This is the cuticle that it will secrete, and this waxy cuticle is what it will prevent the plant from these many things. On the epidermis, be it the upper epidermis or the lower epidermis, in submerged plants on the upper epidermis, in the terrestrial plants on the lower epidermis, we have stomata. Stomata are spores which are present in the leaf. They allow exchange of gases, CO2, oxygen, and water vapor. This is how a stomatal pore looks like. There will be a pair of guard cells, and guard cells do have chloroplasts. Although they are the part of epidermal cell, they are the only cells which have chloroplasts in the epidermis. The opening between them is called the stoma or the stomata. It is partially open. The gases from the intercellular spaces of the leaf can go out through the stomata and just move around by the process of diffusion with the external environment. The next cells that we are discussing are mesophyll cells. Mesophyll cells are of this shape when they are palisade mesophylls. You can see this is the upper epidermis of the leaf. These are the palisade mesophylls. This is the vascular bundle region cut in a different section, and these are the spongy mesophyll cells. You know these are spongy because there are a lot of intercellular spaces there. This is the palisade packed very closely. So both these are mesophyll cells. Mesophyll cells are actually specialized parenchyma. Parenchyma cells are non-specialized packing tissue. We'll come to this soon. Mesophyll cells are present between the upper and the lower epidermis. They carry out photosynthesis because they have chloroplast. The palisade spongy mesophyll, they are actually divided to either palisade or spongy. Palisade are upper, they receive more sunlight, they are closely packed, more chloroplast, and they are elongated in shape. Spongy mesophyll cells are irregular shape. They have the air spaces. They are not closely packed. They have fewer chloroplasts, and these air spaces is a characteristic feature of the spongy mesophyll because it's these air spaces which actually lines up with moisture, and this water vapor here, it evaporates through it just goes out through the stomatal opening, and that's called transpiration, which we'll eventually be discussing, which plays a major role in pulling the water up in the xylem. Collenchyma. Collenchyma are modified parenchyma cells. They have extracellules deposited at the corners for extra strength. So these are the collenchyma cells. You can see that these are just normal plant cell with normal cell wall of cellulose. But the corner, the edges are having extracellulose deposited. This extracellulose at the corners of these cells, it provides extra strength to this tissue. So wherever collenchyma is present, extra strength will be provided to the tissue, to that organ where they are present. And for example, the midrib of the leaf, it's having collenchyma, so that the midrib is actually more stiff than the lamina of the leaf. And not just leaf, the collenchyma is present in the cortex as well. We'll be discussing collenchyma and cortex, and again in cortex of the root or the stem, immense strength is required. So yes, collenchyma plays a very important role there. This is parenchyma. What are parenchyma cells? Parenchyma cells are also just normal plant cells. They do not have any extracellular deposited at the edges. Rather, they have these intercellular spaces. These air spaces are the characteristic feature of parenchyma cells. Parenchyma cells are packing tissue. You can see all these air spaces. So wherever there are air spaces between the plant cell, they can be parenchyma cells, and wherever there are extracellular deposited around the corners or around the edges, that means that those are collenchyma cells. Now, what's endodermis? When we are discussing endodermis, to keep endodermis in mind, we have to discuss some details of the tissue, as in what's present in the endodermis. Endodermis, the cells are one cell thick, and this tissue surrounds a vascular bundle in the roots and in the stem. In the roots, the endodermal cells have two types of cells. They have blocked cells, they have passage cells. So this ring here, actually this star-shaped structure is xylem initially, so you can keep that in mind. And this single cell ring, which I'm just removing my cursor here, you can see this single cell lining of rings. This is the endodermis. And in the endodermis, you can see these are the thickened cells, these are the blocked cells, and these are the passage cells. Again, these are the blocked cells, again, these are the passage cells. Again, these are the blocked cell, the passage cell, the blocked cells, and the passage cell, and so on and so forth. So, the blocked cells, they have extra deposition of a substance which is called as suberin, And this suberin is a tough substance. It blocks the free movement of water into the xylem. And not just water, even the substances which are dissolved in water, for example, any pathogen or any infectious agent, it just cannot move into the xylem because the cell will stop this water from entering. Passive cells, however, they will allow these to reach into xylem or into the root. This is the red shaped region that I have just highlighted here. This is actually your xylem here. And these block cells, I am just encircling them in yellow. So these are the block cells with deposits of suberin. And the passive cells are, I'm just outlining them in purple here. These are all the passive cells. These passive cells allow movement of water. The block cells will not allow movement of water. And these rings, these are the phloem. So in the root, this is the kind of arrangement that we see. Water cannot pass through the block cells if water needs to enter into the xylem. So water will come across the cortex. These are the parenchyma cells. You can see these intercellular spaces. So these are all parenchyma cells. So yes, water will come here, but at the endodermal region, the water will not be able to cross the block cell. They will have to cross only through the passive cell to reach into the xylem. So water enters into the xylem through the endodermal cells, which part of endodermis, the passive cells and not the block cells. So that's what endodermis is doing. And then this is the another tissue. You can just see these cells, which I have not marked with any color here. These are present between the endodermis and the vascular bundle. These are the pericycle. So what's pericycle? Pericycle are 1 to several cell thick cells inside the endodermis and next to the vascular tissue. They are 1 cell thick in roots, and new roots grow from them. Even the sclerenchyma in the leaf, they are formed from the same part. We'll discuss sclerenchyma shortly with xylem. So this is, you can see the epidermis, and this is the cortex, and this is the endodermis, and the xylem and the phloem. And this light shaded part here, under the endodermis and just before the vascular bundle, the xylem and the phloem, this is the pericycle. Xylem, the 3 points, I think we all are very through with this from our very lower classes. They are made from cells joined end to end to form tubes. The cells are dead. The walls of these cells are thickened with hard, strong material called lignin. So what xylem vessels are doing? Xylem vessels in the flowering plants, how do they do? How do they form actually? They are the main vascular tissue for moving water and mineral ions. This is how xylem cells were laid down initially. All the cells were normal plant cells having all the living contents in the cell and the cellulose cell wall. They all were joined end to end, placed one on top of the other. Once they were placed one on top of the other, they were not yet xylem. Lignin was produced inside these cells, and this lignin was deposited in the cell walls. This brown thing is the lignin being deposited. The lignin deposited, it prevents the exchange of any material, no nutrients, no gaseous exchange, nothing can move in or out of these cells. And when nothing can actually move, these cells which were living, they start dying. And when they start dying, the walls, the walls which are present between these cells are removed. All the living contents are removed as well. And you end up getting a hollow lumen altogether. And this is the xylem vessel. So this is how a xylem vessel comes about. Xylem is also having extra lignin rings deposited at different stretches. So you can say extra, this is spiral thickening, this is just stretched, and this is even more stretched. So if you cut through the xylem at a vascular bundle, you can see these xylems, you can see the pits. These pits were initially plasmodesmata. This is the lumen of the xylem, and this cell wall is made up of cellulose now together with lignin. So these are all xylem. Now these cells, these ones are the sclerenchyma. Now what's a sclerenchyma? That's what we are here to discuss right now. So sclerenchyma are quite similar to xylem, but they are not exactly the xylem vessel elements. So what's the difference and what's the similarity? At the outer edges of a vascular bundle, these sclerenchyma fibers are there. These are elongated cells. They have lignin. They are dead. They provide support, the mechanical support primarily. And they are very similar to xylem because they have a hollow lumen, but they are not involved in transportation of water or any ions. They are just hollow, they are dead, they are lignified. That's like it. So this is how these sclerenchyma fibers look like in a vascular bundle, very heavily populated. You can see like they are just clustered all together. And this is the sclerenchyma fibers, and these sclerenchyma fibers, so if you look at them through the lengthwise section or a longitudinal section, this is the lignified cell wall, and this slant here, this wall is, the cross walls are not removed altogether. In xylem the cross walls are absent, so this cell here, it ends in a slant, so that this cell fits onto the other cell, and both the cells have this tapered cell wall at the end. The tapered cell walls with the overlapping arrangements are the characteristic feature of sclerenchyma, and they provide the strength or a good fit for the sclerenchyma to fit in closely together, so that they actually can provide the mechanical support which actually they actually need to do. Phloem. What's phloem doing? Phloem is used for transportation of sugar and amino acids. We know that, and they also end up moving water in phloem, because when the phloem is filled with sugar and amino acid, the water potential gets reduced and water starts to move into the phloem by osmosis. And the hydrostatic pressure which builds up, it provides a mass flow for the phloem sap to move. Two types of cells are present in phloem. The sieve elements, which I am just highlighting these in red. This is the sieve element. You can see the cell wall of the sieve element. I have highlighted this in red, outlined this in red. And the companion cells, I have used this mustard color here for the companion cell. So for every sieve element, there is a companion cell. The sieve element, these cells have degenerate cytoplasm, which means this cytoplasm will be only present on the outer edges of the cell or on the periphery of the cell. The central region of the cell will not have much of the cytoplasm, almost hollow to provide lumen for the transport, and sieve plate and sieve pores are present. You can see the sieve plate and sieve pores here. These sieve pores were initially all plasmodesmatas. Companion cells, so these are also called as the cross walls. Companion cells are supportive cells. They are fully functional. They have all the functional organelles and the cytoplasm which are just present in any kind of a plant cell they can expect. So these companion cells are those. This is the cut section showing to you the cross section of the phloem and the sieve pore. This is the sieve plate. So you can see the sieve elements, and this is the sieve plate you can see, and this is the parenchyma cell, and this is the sieve pores, the plasmodesmatas. If you cut this through this part along its width, so this is how you will see this. This actually shows to you the phloems, and this is again a section of this stem from where the phloem can be seen, and these are the sieve pores. You can see the sieve plates. These have some deposits of this substance, this dark colored substance. What this substance is? This substance is basically the callose. So callose is deposited when phloem sap starts to leak out. For example, if this part of the plant breaks and the phloem sap is oozing out, so the plant will just recognize the high rate of the phloem sap going out of this part. It will seal the nearby these sieve plates with callose, and the deposition of callose is quite similar to the blood clot that we have. It prevents the phloem sap from leaking. It prevents the entry of pathogens. Aphids are insects which feed on phloem sap by inserting their proboscis into the phloem. Phloem sap ooze out too slowly for the callose to form at the sieve plate. So these aphids, they can very successfully feed on the phloem sap, and the callose will not form. Very similar to like the mosquitoes, they keep on feeding on our blood. They take blood meal from us, and our blood doesn't clot because the sting that the mosquitoes have, that sting releases an anticoagulant which doesn't allow our blood to clot. So this was the basic discussion of the tissues that you need to keep in mind regarding the plant's organs. And when we are going further, you will be placing these organs in various parts of the different parts of plants, and you will understand where they are functioning and what function do they add to a given organ. That's it from my side. Thank you so much.